At INRIA in Lyon in France, we were also looking at the virtual modeling of plants. This creates synthetic data sets that can then help us train machine learning algorithms to identify plants in the field or particular traits in real life.
So this is the current bottleneck where we are. We need absolutely massive ground truth data if we want to train this algorithm, this new families of algorithms, to produce these 3D structures and segment it in the correct botanical way. So how to do this? There are two options, basically. The first option would be to acquire real plant data by photograph, by scanner laser, and then that expert segment by hand different parts of the plant, saying that this is a leaf, this part of the point cloud is a leaf, this part of the point cloud is a stem, etc. And you can imagine that this is extremely time consuming, this is extremely heavy task, and we are blocked at this point because of the ability of humans to do such complicated tasks. And there is another option, that is to create artificial plants and then say whether with this segmentation of the virtual plant, whether we are right or not. Because as we designed the virtual plant, we know that this part of the point cloud corresponds to a leaf, this part of the point cloud corresponds to a stem. And because of this, it is possible to automatize the training of the system. So what we want to do in the context of such a phenotyping approach is to use a virtual pipeline where we would produce the virtual plant in the computer. Then we would create point clouds out of these virtual plants. So this would be virtual point clouds. Then we would use training algorithms in the context of this machine learning construction process. And then we would get as an output the trained machine learning system. And then once we have this, it is possible to get back to the original pipeline and use here this trained machine learning system in order to recognize, identify, segment the different organs on the plant. So LPy is the programming language to simulate plants. And from this, it is possible to create full databases of plants by stochastic simulation of plant populations, produce massive data, and then this massive data give them to machine learning systems in order to train this machine learning based on this large amount of input data. The first thing to know is that how plants are growing. You have leaves like this, and then you have a stem like this. This is growing due to this small part at the tip that is called the apex. And the apex is producing all the organs that is being built on the plant. So for instance, the leaves and also the fruits here, or the flowers. So if you look at this in a more detailed manner, if you make a close-up on this, what you observe is that it is like a small dome, like this, that is producing lateral organs, and this is the stem, and these are the young organs, like this can be a leaf, or a flower, or anything else that is produced by the meristem. And this part here, it is the place where all the stem cells are living, and they are dividing, And they are producing the small organs here, one after the other, at the tip of the plant. So if we want to model this, we need to model how this small part of the plant, which is made up of a small amount of stem cells, undifferentiated cells, how this is growing. An apex, let's call it A, is producing a piece of stem that I call I for internode, and laterally it produces an apex that will in turn be able to grow on its own. And we formalize this rule of growth by this, let's say, mathematical expression.
And in the case of ROMI, so this European project we are involved in, we are using virtual plants for a precise objective, which is to use virtual plant to create virtual data set that we can use for training deep learning algorithm very efficiently and costlessly. And as I told you, then once you have an objective, you have to question yourself whether realism and the realistic rendering of the plant is useful or not for your objective. In the program, so we use pipelines that are able to detect automatically, to segment the plant and to detect automatically the organs. Okay, so here basically it will be the branching point that will be discovered, and this branching point can be either branches that are cut here or calyx here. And once it is detected, so we use a representation where we highlight these branching points, so fruits or branches. And you can see that here you have an overlay, and this highlight overlays perfectly with the branches. So you can say, wow, it's really good. But when you dig into details, sometimes this machine learning algorithm, are not working well. And here, when you proceed with the same algorithm, the leaf is recognized partly as a leaf at the beginning, okay, but the tip of the leaf is recognized as a fruit or a branch, or okay. So it shows you here that you have like some issues. So those machine learning algorithms are not perfect, and we need to improve them to increase our performances in terms of segmentation. So I make a zoom in the virtual plant of Christophe, and you can see that definitely the leaves here are in the current model with which we trained this network. They are like quite simple structure with a flat, very simple shape that are not like really the shape of the curl leaf. So here there is an open question. If we make a new plant with modified here leaf shapes, will this help this algorithm to make a better plant segmentation?
To make more realistic plants, we decided to grow real Arabidopsis plants in growth chamber and to measure them in a systematic manner. We then used these detailed measurements to refine our virtual Arabidopsis plant in different ways. First, we refined the modeling of the different organs, for example, the coiling of leaves. With time, the leaf would be produced laterally by the stem in a sort of straight way, like this or like this, and then with time, it would curl and change the shape. Instead of having one single curve, now I will have a set of curves. I would just put the nerve. The nerve would be straight in the beginning. Then I would fold a bit the nerve, fold a bit the nerve, fold a bit like this. Okay, even I can go in this way. Okay. So I will define curve 0, curve 1, curve 6. Okay, actually this is what I did here. And then I say, all this bunch, create me an object that is able, given a time tau, so the curve function object, it is able to take a time tau, and then to return a curve object, a C at the time tau. And then, so this curve object is provided by LPy. You just call it with a series of functions. Then, once you have this, you can call curve of t. And then if I know this t, I can compute the current nerve at time t, and then display it. And actually, what is doing this? It is able to compute all the intermediate curves by interpolation, like this. Detailed models of organs were then assembled to model groups of organs, such as flowers, or, at an even more integrated level, the dynamics of inflorescences development. The difficulty here is to synchronize the growth dynamics of each part. For this, we used a new strategy that we developed in the course of the project, and that is called hierarchical timeline warping. This strategy makes it possible to synchronize the growth dynamics of the different plant parts onto each other in a hierarchical and non-linear manner. Similarly, still based on real plant measurements, we modeled also the observed variability in the dimensions and orientation of the different organs, the gravitropism, and the mechanoperception of the different axes, resulting in complex and dynamic bending of their parts. The final model provides a realistic rendering of the plant growth that can be used as a faithful reference to train Romy's machine learning algorithms. Then, the technology to construct virtual plants developed for Arabidopsis was used to generate other virtual plants with different levels of complexity and accuracy. We first updated our tomato model by introducing stochasticity in the development of the plant. Here, you can observe two tomato plants that were generated using the same stochastic model. The model varies the number of organs, their size, their dimensions, their orientation with respect to their parent stem, their bending, and so on. Another virtual model was made of a chenopodium. This plant is a weed that can be easily found in crop fields. Similarly to Arabidopsis, although with less details, we grew several plants in the growth chamber and observed how it grows. We used these observations to construct a virtual model of a chenopodium. The model is very different from that of a tomato. It grows with a main axis that dominates over a secondary axis that grows in turn with a slight delay with respect to the leader. Here also, stochasticity was introduced in the model so that one can generate different individuals with a simple click. Finally, based on the training course, participants could start to produce their own virtual plants. Here is the first version of a model of a pepper plant, showing the step-by-step approach of the student based on real plant observations. Here is a 3D model of a carrot plant with fractal leaves and stochasticity. Several instances of this stochastic model can be generated to create a virtual field of carrots. So by the way, we will learn on Friday, Ayan will make a presentation showing how we can use these systems and to scan them, make sample of points, 3D cloud sample of points, so that we can train systems, machine learning systems that are that take as an input sample points in order to produce right segmentation of the point cloud. Parts of this video were extracted from a week-long course given in April 2022 and covering every aspect of how to virtually model plants in 3 dimensions with L system in Python. If you are interested, don't hesitate to go and check it out on the Romy YouTube channel.